Good day. We are here to present the Activity 7 Central Dogma. And the members are Manlulu Maylin, Ponce Anime, Castillon Georgie, and Empeno Nicole. And this is the 2D model of central dogma's DNA replication, transcription, and translation.
The process of replication involves copying a double-stranded DNA molecule to create two identical DNA molecules. One of the most fundamental functions of a cell is the replication of DNA. The two daughter cells produced after a cell divides must have the exact identical genetic material, or the DNA, as the parent cell. Each DNA strand already present serves as a template for replication in order to do this. The opening of the double helix and separation of the DNA strands, priming of the template strand, and the construction of the new DNA segment are the three main phases in replication. The DNA double helix, two strands, uncoil at a specific spot known as the origin during separation. Then, a number of proteins and enzymes combine to prime the strands for duplication. Finally, the building of the new DNA strands is organized by a unique enzyme known as DNA polymerase. All cells fall within the broad description of this three-stage process, although depending on the organism and kind of cell, there may be slight changes within the process. DNA polymerase next wraps itself around that strand, attaching new nucleotides to the exposed nitrogenous bases. Once the primer has been placed on a single, unwound polynucleotide strand, the polymerase builds a new DNA strand on the top of the pre-existing one in this matter. The pool of floating nucleotides surrounding the old strand is used by DNA polymerase as it moves down the unwinding DNA strands to construct the new strand. A and T nucleotides and C and G nucleotides always pair with one another due to their molecular shapes, and the nucleotides that make up the new strands are coupled with partner nucleotides in the template strand. Complementary base pairing is the phenomenon that causes the formation of the two complementary strands of
Transcription is
DNA.
the process by which the information in a strand of DNA is copied into a new molecule of messenger RNA, the mRNA. DNA safely and stably stores genetic material in the nuclei of cells as the reference or template. Meanwhile, mRNA is a comparable to a copy form of reference book because it carries the same information as DNA but is not used for long-term storage and can freely exit the nucleus. Although the mRNA contains the same information, it is not the identical copy of the DNA segment because its sequence is complementary to the DNA template.
Translation. Here are the six functions that are here. First is messenger RNA or mRNA. The role of mRNA is to carry protein information from the DNA in a cell's nucleus to the cell cytoplasms. Carries copies of the genetic instruction from DNA to other parts of the cell. Where the protein-making machinery reads the mRNA sequence and translates each three-base codon into its corresponding amino acids in a growing protein chain. mRNA is decoded in a ribosome outside the nucleus to form a particular amino acid chain or polypeptide during translation. Second is ribosomes. A ribosome is an intercellular structure composed of both RNA and protein that serves as the cell's center of protein synthesis. Third is transfer RNA or tRNA, adapter molecules that supply amino acids to the protein synthesis machinery during translation to transform genetic information into protein sequence. Fourth is initiation factors, are proteins that allow RNA polymerase second to attach to the DNA template and begin transcription, as well as an anti-association factor. In the absence of mRNA, it attaches to the 60 subunit and inhibits it from interacting with the 40S ribosomal subunits. Fifth is elongation factor, are the ribosomes workhorses of protein synthesis. They help to lengthen the nascent polypeptide chain one amino acid at a time. In the biological kingdoms, the main biochemical framework of the translation elongation cycle is largely conserved. And sixth is release factors. At the place of tRNA, it binds to the stop codon at the A site. It allows the amino acid to be incorporated to the developing polypeptide by releasing it from its tRNA. Thank you.